I trust everybody can hear me okay. Just to put a little addendum onto Russ's talk, he has written a book on magnetism. I don't know if he mentioned it or not, but we have the book for sale on the book table of the Creation Research Society, and there's no math in it, so it's all conceptual, so you're okay. I work for the Creation Research Society, who is a creation society of biblical scientists and non-scientists who all affirm the authority of Scripture from the very first word. And so we had our board meeting this weekend, and Judy just said, let's come on over and bring some of these board members over here and have this conference. And I think she's done a wonderful job of organizing it, and I know it's frazzling, isn't it? But you're doing wonderful. So, yes. I was teasing Russ a little bit when we were giving some presentations Friday night because I was first, and I said, well, you always start off with the best and then it goes down from there. So this time I'll say, but we make exceptions sometimes to feed the other people, you know, make them think it's okay. So, all right. Dinosaur tissue, the age of the earth. You may or may not be very familiar with this topic, but hopefully in the next 45 to 50 minutes you'll become quite familiar with it. All right, let's introduce the topic of age of the earth just a little bit. In the 1930s, Charles Lyell wrote a book on the principles of geology, and in that he argued that change is very slow. Sedimentation takes a long time, mountain building takes a long time, canyon formation takes a long time, therefore the earth has been around a long time. And he didn't have analytical measurements. You didn't have radioactive measurements, for example, like we have today. So he was making these conclusions based strictly on the idea of he wanted to get Moses out of geology, which means get Genesis out of geology. That was his primary foundation. It wasn't the scientific evidence. In fact, today most geologists will tell you that a lot of Lyell's arguments weren't real, they weren't correct. But the influence it had, incorrect arguments having a bad influence, who would have thought? Okay? The influence it had was dramatic, up to and including that Charles Darwin read his book and said, wow, this now gives me the time for my ideas. And Darwin even stated that half of my thinking came out of Lyell's brain. So what we're really faced with is a biblical timeline versus Lyell's timeline. And why are they not the same? Well, Lyell understood. If you start trying to cram millions and millions of years into the biblical timeline just falls apart. No matter what particular people, theologians, pastors, scientists, whatever say, the truth of the matter is when you start trying to stuff all that time into the biblical account, the biblical creation narrative, it falls apart. And Lyell knew that. That's why he did what he did. We also have to recognize that people will cite it as, well, radioactivity proves this age and it proves that age. Well, first off, the technology to do the radiometric measurements that we think of, rubidium, strontium, uridium, lead, that didn't even exist until the 1950s. Let's count it up. How long was that after Lyell? See, Lyell's arguments of old age, of great lengths of time, have been penetrating and percolating in the scientific community for over 150 years, or almost 150 years. So you see, based upon his faulty reasoning, they had established that this must be 100 million years old, this must be 30 million years old, this must be 10 million years old. Now they develop these radiometric measurements and they start using them. What was their gauge for determining that rubidium, strontium, and uranium, lead was working correctly? If the gauge was, they gave them the date that they anticipated. I promise you, had they been doing uranium, lead testing on certain samples, and they would have come up to ages of 50,000 or 100,000 years, and samples they thought were 30 million years, what would they have done? They said, well, obviously that method doesn't work, we're throwing it out. So you can't tell me that radiometric dating proves the ages. It's a product of the assumption of those ages. There are nearly 500 different physical processes that we can use for determining the age. Only about 10% of those give the ages approximately of what is the accepted ages of today. Why do they toss the other 90% out? Because they don't give the anticipated age that they assume it must be. They assume this layer must be 100 million years. So a process doesn't give us anything close to that age, it must not work. So that brings us then to really two different interpretations of the fossil record, of the geologic column. The evolution interpretation is that the fossils, and this is simplistic, but it's still basically the same. The fossils that are older are going to be toward the bottom. The fossils that are younger are going to be toward the top. The fossils that are older are going to be in older rock layers. The fossils that are younger are going to be in younger rock layers. And what we see then in their interpretation is this nice progression of development over millions of years of time. Voila, the fossils prove evolution. Things have evolved over millions of years. That's how they would interpret the fossil record. Okay, a creationist interpretation is going to be different because we're going to say that this was all laid down approximately the same time by a great flood. But don't let somebody confuse you that the flood still doesn't lay down certain ecological zones. In other words, who's going to be buried first? It's going to be the marine creatures. Obviously, who's going to be buried next? It's going to be the creatures that are closer to shore. Who's going to be buried toward the end? It's going to be creatures up on the higher land. So there is going to be a bit of an ecological zoning that's going to occur. That's what the flood would give you. I've heard many evolutionists try to argue that the flood would give you kind of a washtub effect. They were going to be mixed together. That's nonsense. No reason to assume that. But you see two totally separate interpretations of the same data. We hear also, well, creationists, they don't accept science. That's nonsense. We love science. We just give a different interpretation to the same data. So in the topic that I'm going to address, we're going to deal with the age of dinosaurs, because my argument is that if they've misdated dinosaur fossils, they've misdated it all. Because if the dinosaur fossils are misdated, then all fossils are misdated. And if all fossils are misdated, that means the rocks that those fossils are in have been misdated. All right, dinosaurs are assumed to have lived through three basic geologic ages, the Jurassic, and the Cretaceous, which extend approximately 250 million years ago to about 65 million years ago. So anytime a dinosaur fossil is found, it's automatically assumed to be at least 65 million years of age, and then it goes from there, depending upon other specifications of where it's found. All right, I'm going to directly challenge that. Now, the interesting thing is, I'm not a paleontologist. I've sometimes been referred to as a vocational paleontologist. My background is cell biology and molecular genetics. Say, what in the world would that have to do with dinosaurs? Well, initially it wouldn't have. And if somebody would have told me that 20 years ago I'd be out digging dinosaurs professionally, not for fun, I would have said, okay, there must be, my time continuum must have veered off when I'm on another timeline, because that would make no sense to me, because that's not what I did. But lo and behold, the areas of dinosaur paleontology and cell biology have crossed, and who would have thought? All right, so to address this, the first thing we need to look at is, how do you make a bone? Well, the first thing you do is you lay down a layer of a protein called collagen. Now, collagen is a very tough protein, but it's still biological, it's still a protein. And then you take and you coat that layer of collagen with calcium and phosphorus, which is why your mommy always told you to drink your milk. You need calcium, you need phosphorus to coat the bone layer. You need it for a lot of other things also, but we're just talking about bones here in this sense. Now look, that bone, see, that's not a rock. Has a lot of pores in it. It's actually a living structure. When you're young, your bones get what they call remodeled all the time. And almost every 10 years, you have total remodeling of all your bones. Unfortunately, as we get older and more cranky and more forgetful, our bones forget to remodel also, and we don't have this near as much. Yeah. All right, so the critter dies, the animal dies, the human dies, whatever, gets buried. Have to be buried. That's very important. Need to be buried. Then groundwater enters into the bone, and groundwater brings with it material like silica. And then the groundwater exits the bone and it pulls out the calcium and the phosphorus. And eventually you have what's called replacement, where the living tissue bone has now been all replaced with mineral, and now it is essentially in the shape of the bone, but now it is a rock. And my challenge then is, well, how long does this take? Because what we're going to find out is apparently it takes several 100 million years. Who would have thought? This is such an incredibly slow process. Isn't that interesting? Yeah, it really doesn't. But that's the assumption that's having to be drawn. In 2005, there was a paper published in the journal Science where they detected tissue and fragments of protein still present in the femur of a Tyrannosaurus rex. And then this was followed up very quickly by 2 additional papers in 2007. Now this kind of lit the scientific community on fire because we're thinking, okay, wait a minute, how can there still be tissue and pieces of proteins left in bones that are supposed to be with T-Rex 68 million years old? Now since 2005, there has been a considerable amount of evidence of pliable tissue, stretchable, squeezable, pliable tissue, and fragments of proteins detected in numerous dinosaur fossils located in numerous geologic locations all around the world by several different labs. So it's not just one lab and one fossil in one location. Okay, so this is essentially worldwide phenomenon detected by a lot of different scientists. Also, as part of these additional studies, they've established that even blood vessels can be extracted from some of these dinosaur fossils. And you can find morphologically distinct cells still present in the tissue. Collagen fragments are still found. Remember, collagen is the primary protein in bone. But there's also other proteins in bone, particularly in the blood vessels and other tissue. These other proteins would be like actin, myosin, tubulin. So our challenge is that as creationists, that the presence of intact tissue in these bones, these fossils that are alleged to be over 65 million years of age, is actually a direct biochemical challenge to their surrounding, to their assigned ages, and thus also the surrounding rocks. In other words, the age assigned to the surrounding rocks. So this is one of the reasons that the Creation Research Society is interested in this topic, because this is very powerful evidence that the ages of dinosaur fossils have been misidentified, misassigned. So we have what's called the iDino Project, which stands for Investigation of Dinosaur Intact Natural Osteotissue. And yes, we wanted it to spell iDino. Yeah, that wasn't by happenstance. It didn't randomly evolve that way. All right. You can go onto our website, creationresearch.org, and you can read some about the updates of the project. We also have some other resources on our table that I'll tell you about here toward the end of the talk. We've also published some material, some articles in the scientific literature. Yes, creationists do publish. Surprise, surprise, despite what you're told by the secular world. In 2012, a team of us went up to a region in Montana, digging in a formation known as Hell Creek. And digging this formation, we uncovered a very large Triceratops horn. Now, our museum guides were ecstatic because they thought this might be the single largest Triceratops horn, at least down in eastern Montana, if not the entire state of Montana. But the problem was that we were going to break it up and destroy it to search for tissues and biological material. Thus is the price of science. So, I've also since then found that this picture, which by the way, and it's cut off a little bit, you can't quite see it, but that's my arm on the side there, is apparently very popular on the internet. It's all over the place. Although half of the websites it's found on don't know what they're talking about. I think a couple of them refer to UFOs or something, I'm not sure. It's kind of strange. Mark Armitage was with us at that time doing some of the microscopy, and I was sitting in the back of the pickup truck with the bone kind of half in my lap and half bracing it so it didn't bounce around too much. And it was damp. It had tree root material around it, and it's obvious that from its positioning that it was exposed to the elements, it was exposed to microbes, fungi, and such. And I just turned to Mark and I said, I don't think there's really any chance we're going to find anything. Even as a young Earth creationist, I thought it was just asking too much to have any tissue remaining inside this fossil, even if it was just a few thousand years old, because it had been preserved in such an unideal, such a hostile environment. Let alone try to explain how anything can be preserved in a hostile environment for almost 70 million years. But lo and behold, in some of his work, he pulled this slab of tissue out. And if you notice the dark spots in that, that's cells. It's dinosaur cells. You're looking at dinosaur tissue and dinosaur cells. And this made the cover of the Journal of American Laboratory. If we look at some of this in more detail with the electron microscopy, we see these cells. These are called osteocytes. They are specific bone cells that are found in all vertebrate animals. So that's how we know they're osteocytes, because they're very unique in appearance. Skin cells and muscle cells don't look anything like this. So we know these are osteocytes. And so what you're looking at, these are dinosaur osteocytes. But notice where the arrow is pointing on the picture on your right. Extending from the osteocytes are what are called filapodia. These are very thin filaments of the protein actin. Now imagine, this is a thin filament of a protein. Very, very fragile, still nicely intact after some 70 million years. See, that's what we're being told. This is another fossil. This is a Therosaurus vertebra, or Thescelosaurus vertebra. This end piece was so badly damaged that the museum didn't want it. So they wrapped it in aluminum foil and put it on a shelf. And there it sat for several years. Just sitting there, didn't know what to do with it, just sat there. Well, a couple years ago, a young lady served as an intern in my lab for a little while, and she got her hands on this vertebra because they didn't want it. And she was pulling around and poking at it. And then once you removed some of those slabs, rinsed them off a little bit, this is what you have. So you can throw that on a grill and have a dino steak. You are looking at tissue from a dinosaur. Now, that's clearly a very invasive way of analyzing for biological material. There's also non-invasive ways, so you can't be accused of contaminating or otherwise messing with the sample. You just simply take the intact piece of the fossil and you scan it with, for example, what's called Fourier transform infrared spectroscopy. And it can identify the presence of collagen still in the fossil. In fact, this is not a dinosaur, but a crustaceous bird, which would be the same age as the dinosaur. And lo and behold, this spectroscopy detected collagen still in fossils of this bird. And here's a ceratod rib that's been dated at 195 million years old. Using this infrared spectroscopy, there's the signal. It still contains collagen. Then there's another non-invasive technique called second harmonic generation imaging. It creates a certain wavelength light through the image, bouncing it off, and if it's collagen, it turns red. You see the red spots there. And if we superimpose that onto the fossil, then see, we can see from the Triceratops femur the little pockets of collagen still left in it. Now here's one of the problems that the evolutionary community faces, and that is if you have collagen in these fossils, collagen's at its most stable. In other words, it's most likely to survive long periods of time in this most stable form, which is also its most temperature sensitive form. And any biochemist can tell you the fastest way to degrade biological material, particularly proteins, is to warm it up, cool it down. Warm it up, cool it down. That's why you yell at your graduate students when they forget to put stuff back in the freezer. So in its most stable form, it's also the most temperature sensitive, and yet, and this is not me speaking, this is some scientists in an article in the Science magazine 2008 that say, you know, when these fossils would have been buried in Hell Creek, by their calculations, okay, not Cretaceous calculation, by their calculations, Hell Creek would have been what they call a megathermal environment. In other words, it would have been above 20 degrees centigrade. So that would have accelerated the destruction of the collagen. So therefore they argue that at that temperature, collagen had a half-life of about 2,000 years. Well, if you understand half-lives, that means in 2,000 years you have only half what you had. Another 2,000 years you have only half of that. Another 2,000 years you have only half of that. It doesn't take long to figure out you're not getting 65 million years out of this. Because of that, several biochemists have done studies over the years, and they determined that the most you're going to get of a life expectancy of collagen, even under ideal conditions, and believe me, a fossil buried in the ground is not ideal conditions, not even close to ideal conditions, you're going to get no more than about 4 million years, and probably for the most part you would expect only about 1 or 2 million years under ideal conditions. But remember, they found other proteins also. They found actin, they found myosin, they found tubulin. I can't even get evolutionists to engage me on these proteins. They just want to talk about how tough collagen is. So what about actin? They ignore it. What about myosin? They ignore it because they know that no matter how much they try to argue that collagen could somehow, under some magical, bizarre conditions, survive for tens and tens of millions of years, you'll never get that with actin, myosin, and these other proteins. That's why they won't even touch it. And we're accused of ignoring the data. We're accused of not being scientists. So, how do they determine that collagen will only survive at most 4 million years and typically even less than that? We do what are called thermokinetic studies. It's a protein decay study. In other words, you're going to measure how fast proteins decay. Now, the problem you have with collagen is it is a pretty tough protein. So if I just took a bone and set it on a shelf and said, okay, I'm going to measure the collagen decay in this bone over time, it would be my great-great-grandchildren that would be publishing the paper, because it would take so long sitting on a shelf for the collagen to have any significant decay, because it is a tough molecule. We recognize that. So what you do is you heat the protein up and you accelerate, you speed up the decay rate so that you get to publish in your own lifetime, not someone else's lifetime. And typically the temperatures we're using are 80 to 90 degrees centigrade. Now, it's not Fahrenheit, because I always have to remind my students, there's no such thing as Fahrenheit inches or pounds in a laboratory. You expunge all that. Centigrade, 90 degrees centigrade is going to be almost 200 degrees Fahrenheit. That's how hot we're talking about. Yeah, it'll burn you pretty good. But we can use these high temperature decay rates to then calculate what we would predict would be the decay rate at lower temperatures. In other words, at ground temperature, at subterranean temperatures. And that's been commonly done for decades by biochemists. All right, so the question is, well, how reliable is that? You're heating it way up and you're extrapolating to cooler temperatures. We published in 2015 a special issue of our journal. The Creation Research Society publishes the Creation Research Society quarterly. And we published a special issue that was dealing with some of this and some of the results that I've just showed you. And by the way, we have this issue for sale on our table. A gentleman that works for Reasons to Believe, if you're familiar with that organization, they believe that the Earth is very old, therefore they accept that these dinosaurs are tens of millions of years old. He wrote a book called Dinosaur Blood in the Age of the Earth, and in that he argued that, no, these high temperature decay studies, they don't work. They're not relevant. Now, of course, my first question to him is, have you ever done one? And of course the answer is no, he never has. But that doesn't mean he doesn't have the right to challenge. It just means that he doesn't have the intuitive knowledge that he needs to be able to make that challenge. So, in an article that was posted on the website of Answers in Genesis, I challenged him to provide experimental support for his claim that these thermal decay studies don't work. Said, you're making a lot of assumptions. Where's your scientific evidence to support your assumption? So I got rewarded with 2 articles in response to me, which is fine. And in those 2 articles, he cited a 1972 study that he said shows that at high temperatures, collagen will denature before it starts degrading. And because of that, he argued that that's going to change the chemistry of the decay so that it doesn't apply. Okay? Let me explain to you this in just a second. Here in a second. Okay. If we have a protein, remember proteins, think of a ball of string that's all wound up, it's all kind of knotted up. That's our protein. When you denature, what you're doing is you're in essence straightening out that string, that long string. When you're degrading, you're chopping that string up into pieces. So when you degrade a protein, you chop it up into its individual amino acids. So his argument was that at high temperature, the collagen will denature in such a way that then it's going to change the rate of how it degrades. Therefore, you can't use these thermokinetic studies to predict collagen decay in 68-million-year-old dinosaur fossils. And he claimed that the 1972 study showed that. So he argued that the degradation is going to be faster after it denatures at 90 degrees. Therefore, you're going to argue that collagen can't last more than a couple million years, but that's based on faulty reasoning because the decay studies aren't working correctly. Because he assumes the chemistry has changed once you've denatured at 90 degrees. Well, there's several problems with his argument, and I addressed them in a subsequent article that's posted on the Answers in Genesis website, which, by the way, I work for the Creation Research Society, not Answers in Genesis. We're not the same. People call and say, well, I signed up for your society at your museum. We don't have a museum. It's Answers in Genesis. We've worked together. There were two separate organizations. I know it gets confusing because it seems like everybody and their dog and their cow and their pet giraffe has a creation organization out there. All right? We're the only international Young Earth Biblical Creation Society. Think of it that way. But that also means our members, like the scientific staff at Answers in Genesis, are members of our society. The scientific staff at Institute for Creation Research, including Tim Clary, who will be speaking here later, members of our society. In fact, he's a board member. Rob Carter's a board member. Works for Creation Ministries International. So you have all these different ministries, but the society kind of now becomes the catch pool for all of them because we're a membership organization. So anyway, off of that a little bit. All right, so he assumes the chemistry is different once you heat it to 90 degrees. All right, so what I posted then in response was that, first off, the 1972 study made no such claim about any decay rate differences at 90 degrees versus 20 degrees versus 50 degrees. All right, again, it's just an assumption that this individual made based upon some of the results they published. But the bigger problem was that 1972 study used muscle and skin collagen. We're talking about bone-encased collagen, and it's much different, and it has a much higher denaturation temperature. So the decay rate for bone collagen is going to be much different than muscle and skin collagen. And therefore, his assumptions based on this 1972 study don't apply to the muscle and to the bone collagen because that's not what they studied. And in fact, the thermal decay studies that I cited you used bone collagen. Okay, other studies, as I mentioned, they've isolated blood vessels. They found tropomyosin, actin, histones, tubulin, myosin, all kinds of different proteins found in blood vessels, found in other tissues. Mary Schweitzer, who has been kind of a leading researcher in this field now for quite a few years, and she was the primary researcher on that 2005 paper that I started with. Mary Schweitzer, in an interview a few years ago, said that leaves us with two alternatives for interpretation. Either the dinosaurs aren't as old as we think they are, or maybe we don't know exactly how these things get preserved. Now, she is a very avid evolutionist. She gets mad at us for talking about it. She'd be mad at me right now. Like somehow, see, we're misusing science to draw the conclusion that means the fossils aren't that old. To her, that's a misuse of science. Yeah. But I found this an interesting perspective because she admitted this is where you're at. Either we don't understand how the tissue is preserved or the fossils aren't as old as we think they are. Fair enough. Except here's the problem. Dinosaurs, fossils not being as old as we think they are, that's not on the table. You're not discussing that. So she threw it out there, but that's not really part of the discussion. It's just simply, we need to figure out how this stuff is preserved. Not a matter of, well, maybe they're not as old. No, we just got to figure out how it's preserved. So, this becomes then the question facing the evolutionist community. How in the world do you explain biological materials surviving for over 65 million years, and in some cases over 100, in some cases over 200, and in fact, and yes, I'll give you a plug, Bob. Bob Enyart runs a site where he keeps a list of all this, and there is evidence of biological material and fragments of proteins in fossils that date over 500 million years. Think about that. Now, doesn't it already seem a little more reasonable to assume that maybe they're not that old? See, that's why evolutionists don't like this topic, because the average person begins to already think there's something fishy going on. But they have explanations, because they're not sitting there just saying, yeah, we give up. We can only wish. They have explanations. So one of their explanations we'll call the iron model. They're arguing that iron, the presence of iron interacting with tissue will stabilize the tissue. Okay, in an animal like a dinosaur, there's going to be iron in the hemoglobin in the blood. So when the animal dies, that hemoglobin is going to be released, which is going to release the iron. And they're going to argue that there's high concentration of blood in bone. We may question that, but that's what they're going to argue. So they're going to say that the blood always going to work to preserve the biomaterial. All right, here we go. A little bit of chemistry, no math, just chemistry. All right, this involves what's called fitting chemistry. The iron will catalyze. When I say catalyze, that means it's going to initiate a bunch of reactions. They're going to result in the formation of some products, including some ions and also what's called a free radical. In this case, a hydroxy free radical. If you remember in your chemistry, ions like to seek their opposite charge. So it's a negative charge ion likes to seek a positive charge ion. But a free radical doesn't care. It just reacts with everybody and everything. And that's why free radicals are very dangerous to living organisms like us, but they also react with material in a dead animal, such as a dinosaur bone buried in the ground. The free radical, like in this case, the hydroxy free radical, will initiate what are called cross-linkings. Let me illustrate that. See, the free radicals will react with the amino acids. Each of these circles is an amino acid making up the protein. They'll react with two amino acids in this illustration in such a way that the two amino acids will now cross-link. Think of it as taking a string and just starting to wind it tighter and tighter and tighter. The argument is then that if we get it tight enough, cross-linked enough, that it now becomes much more resistant to microbial attack, much more resistant to enzymatic attack, therefore it can survive somehow in the ground for tens of millions of years. Okay? That, in a nutshell, is the iron model. Now, there's only been one experiment to try to validate this, and it was not that great an experiment. We'll explain what they did. In 2014, Mary Schweitzer, I just quoted, she and her research team, they did a study with ostrich blood vessels. What they did was they soaked the blood vessels in either hemoglobin or water. And then they analyzed the degradation differences of the water-soaked blood vessels and the hemoglobin-soaked blood vessels. And what they determined is the water-soaked blood vessels were suffering significant decomposition after just 3 days, where the hemoglobin-soaked blood vessels were still intact after 2 years. So they said, voila, look at this. We got a 240-fold increase in stability, which then they calculate out, well, if you're saying we're going to get 2 million years from collagen not in this condition, then we can get, 240 times that by this method. And therefore that covers everything and explains everything. And this has been considered the primary explanation. And in fact, in the movie Jurassic World, yeah, we now know tissue gets preserved by iron. Next question, it's all answered, it's all taken care of. Well, that's one of the reasons the Creation Research Society is doing its iDino project, because we don't want the evolutionists to set the narrative and control everything that's being said and everything that's being studied. We come along and we ask the questions that they really don't want to ask. And one of those questions is, okay, what's really going on in this study? Well, here is black and white pictures of her results. There's the hemoglobin soaked picture of blood vessels on the top and the water soaked pictures of blood vessels on the bottom. And you say, wow, boy, it really is. It's a whole lot better preserved. Well, as Paul Harvey would say, now for the rest of the story. Okay, here are a few challenges. First off, 2 years, well, not trivial by any means. Certainly, you can't just extrapolate it to 70, 80, 100, 150 million years. Big difference. Especially at fluctuating temperatures as opposed to your nice, stable humidity and temperature in the lab. The second thing, the study admitted, catch this, they admitted that they could not get sufficient release of the hemoglobin from the red blood cells. So they had to physically disrupt red blood cells to get sufficient hemoglobin into the sample. So by their own admission, this doesn't actually work. So apparently the dinosaurs carried blenders around, and when they died, they blended themselves up to make sure there was plenty of lysing of their own red blood cells. All right. Also, most dinosaur skeletons are what we call highly disarticulated. In other words, they're widely scattered. It's a bone here, it's a bone here, it's a bone way over there. And that's why if you find a dinosaur skeleton that's mostly intact, 70, 75, 80% intact, you're a millionaire. Because that's so rare, museums would pay so much for that. Yeah, think about it. If most dinosaur fossils were widely disarticulated, what that means is they were broken up as part of the burial process, which also means they just didn't die down and were buried. Okay, they were broken up. But that also means that these bones weren't buried in a bloodbath. So already you have a bit of a challenge of, do you even have sufficient hemoglobin present to give you enough iron to make this work? And another problem, water washes out the color. Remember the color of the water soap? It was nice and clear. While color is hemoglobin when it's exposed to oxygen, yeah, bright red. So let me see. I'm going to take blood vessels and soak them in hemoglobin and get them exposed to oxygen and then take a picture of them. And then I'm going to let water soak through the blood vessels for a few days and take a picture. And oh, these are nice and red and they're nice and healthy, and these are all pale and anemic looking and yuck, and so obviously it's better preservation. Grocery stores know this. That's why they make sure that the meat is nice and oxidized so it looks bright red. If you have a bright red piece of steak and a brown piece of steak, which one do you buy? They'll taste just the same, but they know, oh, that red one's always look better. So I would challenge that in Dr. Schweitzer's work, immediately there's this subjective mechanism going on in the mind. Well, the hemoglobin cells want to look healthier because they're nice and bright and red, and they just look better. But here, to me, is the biggest single weakness of this study, and that is the results were all just visual analysis. In other words, they didn't do any chemistry. They didn't do it analytical. They just looked. Ah, looks better. Looks better. They declared, for example, that the water-soaked blood vessels had greater deterioration of the blood vessel wall. And my challenge was, how do you know? You did not describe any protocol for measuring or comparing. Grossly unscientific. They just looked. Just looked. You don't make quantifiable conclusions without quantifying. I mean, it's just that simple. That's third grade. You don't make quantifiable conclusions without quantifying. You don't say, it's 240 times more effective without measuring the effectiveness. I would challenge that they maybe didn't want to look too much closer. You've heard the little saying, don't look the gift horse in the mouth. For those of you who didn't grow up on the farm, see, you can determine the approximate age of a horse by its teeth. And so that don't look a gift horse in the mouth is based upon somebody gave you a horse, and then you look in the mouth and you realize this horse is 35 years old. Not much of a gift horse after all. I would say that evolutionists are very reluctant to look this gift horse in the mouth. They don't want to dig too deep into the lack of analytical sophistication of this study. And as I say, this is the only study that's been done to verify the iron model. Also, it's been a situation where, and this again is not the creationists saying this. This is in other publications by other scientists that aren't creationists. There's not enough iron in the blood. You just simply don't have enough. Therefore, you're going to have to get iron from the rocks, the geologic sources of iron. The problem with geologic sources of iron is they're highly insoluble. You're going to have to use water, just like you use water to bring the silica in to make the fossil, you have to use water to bring the iron in to preserve the tissue. But the geologic sources of iron don't really solubilize in water. In fact, they are very insoluble in water. In fact, it generally requires an extremely low pH to get them to solubilize. We've done testing in my lab, and the only way to really get geologic sources of iron to be soluble in water enough that the water can transport the iron in is we have to have a pH of 1. What else has a pH of 1? Hydrochloric acid. Now, that's going to preserve your tissue real well, yeah, isn't it? Right. So now it's a matter of can I get enough iron in to preserve the tissue before the acid eats the tissue up? It's also a matter of if I'm very slow transporting the iron in, is there going to be any tissue left to preserve by the time I get all the iron in? And this is a conundrum. Now, you won't hear this on National Geographic, you won't see this on Discovery Channel, but in the scientific literature, this is a conundrum. We can't figure out how to get the iron to work. Even if the chemistry does work, which I challenge it does, but even if the chemistry does work, we can't get enough iron into the fossil. So the model is kind of dead. There's also another model that's just been proposed here in the last few months, and we call it the burnt toast model. Okay. In this model, they are analyzing oxidation products. These are called advanced end products of glycolysis and advanced end products of lipolysis. So in other words, there are certain chemical states of carbohydrates and certain chemical states of lipids. So they call them AGE and ALE. The presence of these oxidation products gives a brown, burnt color to the tissue. We call it the burnt toast model because when you take a piece of bread and toast it, you are generating these end products, and that's why the toast is brown. Or black if you left it in too long. There's other reasons for the color as well, but these are contributing. So that's why we call it the burnt toast model. All right. What they're arguing is that these chemicals will surround the tissue and give a protective layer. It'll also give it, like the iron model, give an intense cross-linking. So what they're saying is that the wavy lines are the tissue, then you coat it with these end products. So now what happens is, that water is now reflected out. It's very resistant. The tissue is now resistant to the presence of water percolating in, which means you can't get the iron in there. So they seem to be kind of contradicting. But anyway, also microbes won't be able to penetrate very well. So the tissue now is protected from both water and from microbes. And that's the thinking behind the burnt toast model. Again, some challenges. Well, first off, not all dinosaur tissue is brown. But yet, if you're going to have these end products, they should be brown. You know, I don't know about you, but in the restaurant, if my toast comes out and it's still white, I send it back. It's not toasted. All right. Also, cross-linking, both in the iron model and the burnt toast model, you have this intensive cross-linking. It's not going to be stretchable. You lose the stretchability. Also, this alters the amino acids. It's converting them to these end products and other types of products. Well, how can you analyze to say you have these amino acids there to connect the protein if the amino acids have been chemically altered? There's been no experimental verification of this method. It's just simply a speculation that, well, that's good enough. And one of our challenges is how in the world are you able to argue that these natural polymers of these AGEs and ALEs last longer than plastic. Because you won't put plastic in the ground and it'll still be there 200 million years from now. I may tell you it'll last a long time, but it doesn't last that long. But apparently these chemicals do. All right, I'm going to close then with a quote from a geochemist named Jeffrey Bodden. Now, Jeffrey Bodden is very much an evolutionist. I don't want to in any way suggest that he's somehow on our side. He is absolutely not. But he was quoted then as challenging that he cannot imagine that this tissue survives for millions of years, because he says, even if the T-Rex had died in a cooler, drier climate than Hell Creek, in other words, even if the T-Rex fossils, and this is before the other fossils, so we can even broaden it and say, even if all these fossils were found under more ideal preserved conditions, environmental radiation would have degraded its body. Bones absorb uranium and thorium like crazy, thorium like crazy. You've got an internal dose of these radioactive materials that would wipe out any biomolecule. I cannot get a single evolutionist, including the one that works for Reason to Believe, to engage me on this topic. They just don't want to talk about it. And here it is. Even if you have all of these other ideas of how it could be preserved, there is no model even close to accounting for surviving 60, 80, 100 million years of exposure to radiation. You simply can't do it. That's why they won't even talk about it. So yes, we're going to say that the dino tissue is consistent with the biblical age of the Earth, but it's very inconsistent with the standard assigned ages of the Earth. That's why we do not accept that dinosaur fossils are millions of years old, which means that no fossil is millions of years old, which means that no rock is millions of years old. And with that, I point then some resources we have on this topic. There's both a book and a video called Echoes of Jurassic, as well as that quarterly that I mentioned. And I think we have just a couple or 3 minutes maybe for questions.
Yes, we do. You turn on this mic. Does anybody have a question? Welcome to come up here.
They're all stunned.
Hello. My question for you is these dinosaur tissues that we have found, if we're assuming the flood, then if we look at those guys, do we think they're all about the same date? Because then they kind of should.
Right. Yeah, we would assume that the dinosaur fossils we are finding were all buried during the flood. Yes.
I don't know how you're going to reduce time to this, but when I was a little boy, growing up in North Dakota, my dad and the other farmers and ranchers used to go into the Badlands and dig their own coal. And my dad was very good. I was just about this big ears, and my dad would drill into the side of the buttes, and he'd get to the seam of the coal, put the dynamite in there. It'd be generally about 6 or 7 things of dynamite, and then he'd say, run over there and get their heck out of the way, because that thing is sliding out. And when that went off, that big vein, which would be about the size of that, comes sliding out of the side of the mountain, or not the mountain, the butte. Well, anyway, the dad, then he started his work of splitting the veins. But he showed me many, many times. Because I was just a little fellow, and I went with him all the time. Every evening, in fall. And he'd split these that are coming out of there. And here you would see hundreds of little snails, they weren't alive. All different types of things that you could find and slide out. And I would ask Dad, where did these come from? Dad said, he just pointed up, he said, there's the answer up there. And to me it was just incredibly exciting. And I can show you buttes right now in Williston, North Dakota, that I have in my home. I got a stone about the size of that table that's got all the ancient things in the veins. It's available for anyone who wants it, but it's I don't want it anymore. It's big. But anyway, I hear your conversation and think of the wonderful way that you can learn something of these veins that comes sliding out of the side of the hill of you, and it's got snails in it, it's got little birds, veins, and the fish of the size of your fingers. Hundreds of these come out there. Well, that's about it. I don't want to spend more of your time.
Thank you, and let's wish Vern a happy birthday. Is it 90 today? Is that right? 92. 92 years old. Okay, one more question.
Yeah, obviously the presence of all that extraneous material in coal beds would be much more consistent with the rapid simultaneous burial as the creation model, the flood model would predict. Yes. Okay.
But have you heard the original story, the femur that came out of the Hill Creek Formation that Mary Schweitzer found the blood vessel?
That was the 2005 report, correct?
That actually dropped from the helicopter out of the
Well, what happened was that it was so big that the helicopter couldn't lift it, so they had to intentionally break it in half. And I'm sure that hurt. I'm sure they didn't want to do that. And that's when she then went to Jack Horner, who was her ranger professor, and she said, well, now that we've broke it, can I take some of this interior sampling? Because her background was more histology. And he said, sure. And that's when she began to look and say, you know, I'm seeing stuff here that looks more biological than mineral. You have to recognize also that her 2005 report was not the first. It was just the first one that really caught attention. So there had been reports of some kind of biological material in dinosaur fossils dating back into the 60s, if not even before, but it was just generally dismissed as contamination, mistake, whatever the case.
That's the current story, that they broke it, 2,000-pound femur, but there was an earlier story out there that it actually fell out of a sling, and I think God, when I learned about that God broke it on purpose. How do you break a 2,000-pound femur, and would they do that? Because anyway,
So,
I wonder
I can't not say that it wasn't an accident, but they tell you that they did it on purpose because the helicopter couldn't lift it.
This is sacred.
Yeah.
Okay.
So we're assuming that all these dinosaurs will lay down. We're assuming that all dinosaur bones will lay down in the same period of time.
Laid down during the flood, right, not exactly at the same time or exactly the same place, but certainly within the time frame of the flood, correct.
So
Yes.
are there bones being analyzed that don't show soft tissue? Or
Oh sure, yeah. It's not like every single bone has soft tissue, but most dinosaur bones also are not examined for soft tissue. It's not something you routinely do, especially if it requires breaking the bone up, which they're not necessarily anxious to do.
But there's an explanation that some aren't.
Yeah.
Soft tissue.
Right. There was in a few years ago, there was an article in the journal Nature Communication where the authors simply referred to the soft tissue as a common phenomenon. In other words, even by then, a few years ago, they determined that it's frequent enough and widespread enough that we're not surprised anymore to find tissue in a fossil.
I don't know who to address this question to, but I'm wondering if that meteorite iridium layer theory still big now?
You're talking, okay, the better person to address that question to is Dr. Tim Clary, who'll be speaking here later on. He just loves talking about that stuff.
Okay, thank you very much, Doug.